So next would be the start of cytoplasmic organelles. First one is endoplasmic reticulum. Endoplasmic reticulum is a membrane-bound cytoplasmic organelle. It's a membrane-bound cytoplasmic organelle found inside cytoplasm. So regarding its structure, endoplasmic reticulum is a network of flattened sacs and tubules. So let me draw over here. What does this mean? Flattened sacs are called cisternae and tubules are tubular in structure. So these two components together constitute endoplasmic reticulum. So how these two structures are arranged, let me draw a diagram. This is a flattened sac and we call it as cisterna. It's a one cisterna. So this one cisterna is connected with another cisterna with the help of a tubular structure. I will just use another color, and this is a tubule. So this tubule, it connects two cisterna, one and this. So in this manner, a number of cisterna, they are placed one above the other and are interconnected by tubular structure. And this pattern, it forms the endoplasmic reticulum. In this way. Again, endoplasmic reticulum is a interconnection between cell membrane and a nuclear membrane. If I draw over here a boundary of a cell, this is the cell membrane because phospholipids molecules are over here. So this is a cell membrane. One part of cisterna or endoplasmic reticulum is linked with the cell membrane, and its another part is linked with the nucleus. So these are the two membranes of a nucleus. This is interconnected. So endoplasmic reticulum is the cellular endoskeleton. It connects cell membrane with the nuclear membrane. So it serves as a passage for the molecules to pass from cytoplasm to nucleus and from nucleus to outside into the cytoplasm. So this is regarding the structural aspect of endoplasmic reticulum, that is made up of cisterna and tubules. Let's see the diagram. It's a better explanation. It's give you a better overview. Regarding structure, it is a network of cisterna, tubules and vesicles. What is a vesicle? In this diagram, a cisterna is the site for synthesis of two components, one proteins, so another steroidal hormones or steroids. So these two components, when they are synthesized from the interior of the cisterna, from the space from the cisterna, these are packed into vesicles. Say protein is synthesized and protein would be released into the vesicle. This protein molecule is relieved from cisterna and it passes through the cytoplasm and enter into Golgi body. And this movement of vesicles containing proteins and steroidal hormone from endoplasmic reticulum to Golgi body is called cytosolic trafficking or movement of vesicles. So endoplasmic reticulum, based on structure and function, is of two types. One type we called rough endoplasmic reticulum. This endoplasmic reticulum, it contains ribosomes on the cisterna. So their function is synthesize proteins. As in this diagram, these red colored circles indicate ribosomes. It's a rough endoplasmic reticulum and synthesizes proteins which are packed into vesicles, and these vesicles transport to Golgi body for maturation. This is cytosolic transport. So if a cisterna is without ribosome, we call it smooth endoplasmic reticulum. Its function is to synthesize steroids and steroidal hormones. So on the basis of structure and function, there are two varieties, two types of endoplasmic reticulum. Just look into this diagram. Here is the nucleus part, and this nucleus part is surrounded by nuclear membrane. These are all cisternas. This is one cisterna, this is tubular part, and this is next cisterna. These two cisternae are connected by tubules. These pink colored dots indicates the attached ribosome. So this is structure of rough endoplasmic reticulum. So this structure shows tubules and cisterna. These are without ribosomes. So these are the smooth endoplasmic reticulum. So another cytoplasmic organelle are the ribosomes. Ribosomes are called as protein factories of cell. Ribosome is the only cytoplasmic organelle which is without cell membrane. And about the structure of ribosome, a ribosome is made up of two components. Its one component is called large subunit. Its another component is called small subunit. This is large subunit. This is small subunit, and both are linked by messenger RNA at the time of protein synthesis. These two parts come together, and as we can see in this diagram, this part is the large subunit of ribosome, small part is the small subunit of ribosome. During protein synthesis in cytoplasm, small subunit binds with the messenger RNA, then large subunit attaches to it. Large subunit contains three important components. These are designated as E, P, and A. These are three sites on large subunit. Like if we call it as A site is for attachment of new amino acid. P site, where polypeptide chain elongates and it grows, like this is a polypeptide chain. And E site is a site from where the transfer RNA leaves the large subunit of ribosome. So generally, two subunits of ribosome, they remain scattered in cytoplasm. During protein synthesis, these subunits associate together on the surface of ribosome. And what's the function of ribosome? These are with the function of protein synthesis. So another is the cytoplasmic organelle we call mitochondria. Mitochondria are called powerhouse of cell These are concerned with synthesis of ATPs, or which is the energy currency of cell. Regarding its structure, a mitochondria has double layered membrane. Second, it has the outer layer and inner layer. Its outer mitochondrial membrane, this is called inner mitochondrial membrane. Third point, inner mitochondrial membrane is thrown into finger-like folds we call invaginations. These finger-like folds are called cristae. Each cristae has head, stalk, and base, three components a cristae. A cristae contains enzymes for electron transport chain, which is concerned with ATP synthesis. Fourth component, inner to mitochondrial membrane, mitochondria contains matrix, which has double stranded DNA, which is circular in nature, plus it contains enzymes for beta oxidation and regarding function, I said it is the powerhouse of cell. Let's see the diagram. This diagram depicts clearly the parts of mitochondria. So this one is the outer mitochondrial membrane. This is inner mitochondrial membrane. It gives rise to finger-like outgrowths or folds. So this is crista, one crista. It bears these elementary particles, and elementary particles, they contain enzymes for electron transport chain, and it has a double-stranded and circular DNA. This is mitochondrial matrix, and these are the ribosomes which are present in mitochondrial matrix. So this highlights of structure, they repeats the same thing as the function. It is a source of energy for all body functions. So another component is Golgi body. Golgi body is made up of a number of flattened membrane bound sac-like structures which are called cisterna. Here there is absence of tubules. So these cisterna are placed one above the other to form a pile of cisterna or a stack of cisterna. Just like the diagram, just I'm going to draw over here. So this is a pile of cisterna. And if I arrange them in this pile, from each cisterna, vesicles are given off. These vesicles contain mature protein and steroidal hormone. So when the cisterna are arranged one above the other, there are three regions are formed. The regions of cisterna which faces towards endoplasmic reticulum is called cis phase of cisterna. And a phase of cisterna which is in the middle of a pile is called mid phase of cisterna. And the phase of cisterna, a pile of cisterna which is towards the cell membrane is called transphase. So the vesicles containing immature protein and steroids, suppose it's a vesicle, it has immature protein which is synthesized by rough endoplasmic reticulum, says it is insulin. It is immature protein. It is carried by this vesicle. It enters the Golgi body at cis phase, delivers the immature protein. Immature protein undergoes post-translational modification, and finally the mature protein or insulin accumulate in the transphase, and from the transphase it will be loaded into secretory vesicle. This is secretory vesicle, and this vesicle carry this insulin molecule towards the cell membrane. Say it is, I am drawing over a cell membrane. This is a nucleus, and say it is a beta cell. So this is a secretory vesicle which carries insulin molecules. This secretory vesicle, it fuses with the cell membrane, and the insulin will be released from the cell membrane. So in this description, I have provided three functions of Golgi body. One, immature insulin vesicle enters this phase and it undergoes maturation. So one function is Golgi body brings about modification or maturation of the proteins and steroidal molecules. Second, matured protein molecules, there are different protein molecules which undergo maturation, may it be enzymes, may it be hormones, may it be structural protein. So all the proteins after maturation, they are loaded into different secretory vesicle. So second function would be sorting of molecules. One is modification. It carries out the modification of molecules. Afterward, these are called sorting of molecules, mature molecules. And third one is called transportation of mature molecules to their destination. Here in this example, if it is a beta cell, we have assumed, so this is a secretory vesicle which carries mature insulin. It fuses with the cell membrane and insulin comes out of the cell membrane, and this process is called exocytosis. So a beta cell performs three functions. So a Golgi body in the beta cell performs three functions. First, modification of immature insulin. Second, sorting of insulin in secretory vesicle. And third, transport of secretory vesicles to bring about a release of insulin. So these are the three functions of Golgi body. This is a diagrammatic representation. So in this diagram, again this is the nucleus, this is the rough endoplasmic reticulum, and this is a Golgi body. Golgi body has a number of cisterna placed one above the other. So this is the face of cisterna which is towards endoplasmic reticulum facing nucleus, is called cis phase. This phase receives vesicles containing proteins and steroids from endoplasmic reticulum, may it be rough or may it be smooth endoplasmic reticulum. So these components, proteins or steroidal hormone, are poured into the interior of cisterna where they undergo maturation, and ultimately matured molecules comes to the level of transphase. This is the transphase which faces towards cell membrane, and this is the matured protein molecule released from the interior of cisterna. It is packed into secretory vesicle, and a secretory vesicle, it fuses with the cell membrane, brings about the release of matured protein or steroidal hormone. So the functions of Golgi body are three: brings about modification, brings about sorting. Sorting means separation. Separation of matured enzyme from matured hormones, from other structural protein or from steroidal. And then they are packed into different secretory vesicles which are transported to the destination of those molecules. So another cytoplasmic organelle which performs important function in the body are lysosomes. A lysosome is a membrane-bound small vesicle or bag-like structure. It is the membrane, and interiorly it contains enzymes. These are the enzymes. So these enzymes, since these are in lysosomes, so these are called lysosomes. All the enzymes in lysosome, they belong to the category of hydrolases. And hydrolase is a class 3 enzyme. When we study the classification of enzyme, which are of 6 types, one is oxidoreductase, second transferases, third hydrolases, fourth lyases, fifth isomerases, and ligases. So hydrolases are those enzymes which split a macromolecule by addition of water molecules. So example of hydrolase is lipase. Lipase split triglyceride into two components. One is glycerol, other is fatty acid. So all the digestive enzymes are hydrolases. So what is the function of lysosome or lysosomal enzyme? Lysosomal enzymes, they bring about breakdown of excessively stored molecules in body like glycogen, like mucopolysaccharides, like the bacterial toxins. So all these components, when they are in excess or toxic to the body tissues, these are degraded by lysosomal enzymes. So the lysosomal enzyme function stands for removal of waste toxins from the body. It brings about the decomposition of the dead cytoplasmic organelles, and it brings about the phagocytosis and destruction of pathogens in human body. Next, most important part of a cell is the nucleus. A nucleus is the regulating component of a cell. A nucleus controls all functions of cell. Nucleus controls cell divisions. Nucleus control cell differentiation. Nucleus control repair of body tissues. Now, since nucleus is so important, now let's discuss the structural part of a nucleus. First of all, nucleus is surrounded by double layer membrane. These are outer nuclear membrane, and this is inner nuclear membrane. These two membranes, they are not completely separated because these two nuclear membranes, they join together at nuclear pores. So they remain separated. Second part, internuclear space contains fluid. Third component, nuclear pore. A nuclear pore is a channel in the nuclear membrane through which transport of molecules take place. The molecules from cytoplasm can enter inside nucleus, and the molecules from nucleus, they can migrate towards the cytoplasm through nuclear pore. Then fourth component, inside nuclear membranes, there lies nuclear sap. It's a fluid that contains different components of nucleus. Fifth one, nuclear sap contains chromatin material, which is the genetic material of a cell. This nuclear material is made up of two components. One is DNA, deoxyribonucleic acid, plus it contains histone proteins. These two components, that is a nucleic acid with a nucleoprotein, together constitutes chromatin material. Chromatin material during prophase loses water and become condensed and is appear in the form of chromosomes. In human body, there are 46 chromosomes. So each cell of human body is said to be diploid. Diploid means total chromosomes are arranged in one pair. That is 23 chromosomes. They pair with another set of 23 chromosomes. So there are a pair, two sets of chromosomes. So each cell of human body is diploid. Let's see the diagram. This is a good diagram of a nucleus. So it shows this nuclear envelope. This is the outer one is outer nuclear membrane. This is inner nuclear membrane. And these two nuclear membrane, they fuse together at this place we call nuclear pore. It's like this. If I draw over here, this is how the diagram represent. So this is outer nuclear membrane. This is inner nuclear membrane. So there is, this one is intermembrane space, and this part is the nuclear pore. Nuclear pore where the nuclear membrane separate. So this is the site for transport of material from cytoplasm to nucleus and outside. And inside this lies the chromatin material. This is chromatin material. And over this, I can draw over here a nucleolus. So these are the components of a nucleus. So regarding functions, earlier I said in my previous slide, it controls cell function, controls cell division, cell differentiation, repair of body tissues. And nucleus is the genetic material because nucleus carries information from parents to offspring.